that's come out from essentially every single TCGA or other genome sequencing paper. And these are just screenshots from 4 different such papers, making the point that if you ask, where is the cutoff for mutations that are present in 50% of the tumors, and sample size your project to be able to collect all mutations that are present at least a 20% frequency, you see that only 2 mutations, or even 1 mutation in the case of ovarian cancer, is present in 20% of cases. So it's absolutely clear that we have to lower our threshold for mutation frequency, increase our numbers in order to capture many of the mutations present in these tails. And why is this so important? Do we really need to know every last detail? Well, I think the ovarian cancer is extremely telling in this regard, because at least 4 druggable or potentially druggable driver mutations are present at a less than 2% frequency in ovarian cancer, and there they are. So I was quite interested in a publication that came out in January in Nature by Gadi Getz and colleagues at the Broad Institute, who took a statistical look at over 5,000 tumors in various databases on which whole exomes were available to think about this problem. And I want to review one conclusion from that and then take it to another level. So this was a saturation analysis of looking at 21 tumor types, roughly 5,000 tumors. And if you use that threshold of are there still more genes to discover that are present at a greater than 20% frequency, the answer is probably no, and that's shown by this red projection. If you were to go to sequence lots more patients, this line looks like it's flat. But in every single other curve shown here, in which frequencies are lower, you clearly gain much more knowledge by sequencing more patients. The estimates in this paper to get 90% power to detect 90% of the genes at a frequency of greater than 2% are you need 2,000 samples. If you were to do that for lots of tumor types, let's say 50, you need 100,000 tumors. So I'm not showing you this to tell you that I want us to spend NIH money on sequencing 100,000 more tumors. I want to make a few other points